This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
My name is Marieke Krudering-Hol. I'm an associate professor here in the Department of Pharmacology. I did all my training in the Netherlands, where I'm from. I studied biopharmaceutical sciences, then did my PhD in toxicology, all at the University of Leiden in the Netherlands. Then I did my postdoc in cancer research at the Imperial Cancer Research Fund in London. And then when that lab moved to San Francisco, I moved to the Cancer Center here in San Francisco. And while I was doing my postdoctoral research, I got involved in teaching, and I enjoyed it so much. And they were looking for somebody to teach with a pharmaceutical background, so that worked out quite well. And since then, I've been dedicating myself full-time to teaching our students here in the medical school, the pharmacy school, and the dental school. And it's enormously enjoyable, and it's great to work with wonderful colleagues here. And I'll do my best to keep on doing it tonight. So that's a little bit about me. And let's just go ahead and continue then our series following up on where we started last week. So the title of tonight's session is Bugs and Drugs, Part 1. And of course, given that I am in the Department of Pharmacology, which is the study of the interaction between living things and chemicals, the living things often being us, human beings, and the chemicals, the drugs we take, of course my focus will be more on the drugs. But let's recap for those of you who weren't here last week. The bottom line is last week we learned that the immune system is extremely important, and if you don't have one, you won't be able to live and you'll turn into dust pretty quickly. The immune system is very complex, lots of little bits and pieces involved, and it's truly awesome in its capacity to defend us from microbes. We also learned that we have lots of bacteria on our outside, so on our skin, in our oral cavity or our mouth, and in our gut, and that will be important to what we will be talking about tonight. And Dr. Gundling introduced you to some of the names of some of the invaders that we have to keep at bay if we're lucky with our immune system alone, but if not, we can always get some help from the pharmacology department. And some bug names that were used last week were anthrax, when we were talking about the scare of somebody sending around envelopes with white powder. E. coli was mentioned, pneumococcus, and H. influenza, and Staph aureus. And again, you may remember the video of the little thing that was finally eaten up by the neutrophil. That little tiny speck was the bacteria Staph aureus. And in the field of pharmacology, this is actually quite a problematic organism. And we'll talk more about that tonight. And so we also learned that sometimes the immune system has trouble, and this can be genetic and quite severe or debilitating or having a great impact on life. And we met Elizabeth, who told us her story, and in her case, the immune system had trouble making antibodies. But you learned the immune system has B cells and T cells, and so you can have also problems in your immune system with just the T cell side of things. And for instance, if unlucky individuals get infected with a virus causing HIV, then your T cells will be affected. And we will talk about what we can do pharmacologically or using drugs for this second condition next week, because there's a lot of interesting drugs and a lot of interesting science involved in managing this. But today, let's go back to, so sometimes the immune system has trouble. And in fact, we may not have CVID or the condition that Elizabeth was suffering from, but I bet even us, all being healthy and not suffering from the condition Elizabeth has, my question to you is, did your immune system ever have a little trouble? Or the other way of asking you that question is, did you ever need to take antibiotics? And in the modern world, it is pretty hard to find an individual who throughout his or her lifetime never, ever took antibiotics. So does somebody think that maybe they never? So as we can see, many of us at some point in our lives had an immune system that needed a little help. And when your immune system has trouble fighting off the invaders, drugs can really, really help. And this is an understatement, because since the invention of antimicrobial drugs, millions and millions of lives have been saved, and much more invasive surgeries have been made possible because of our understanding and use of antimicrobial drugs. So of course tonight we're going to talk about what kind of drugs are they and how do they work. But we are also going to sort of give you some answers to some of those practical questions. Like often you will hear people get treatment with antibiotics and then they end up with diarrhea. Well, why is that? And tonight we'll find out. Also, the doctor may give you a series of prescription for, let's say, 14 days. You take your pills for 6 days, you already feel better. Why should you keep taking those pills? Because you're better, so, why go on for 14 days in total? And again, tonight we will find out. And also, if you paid close attention, one of the things Elizabeth mentioned last week was that sometimes, she would have infections, coughing, and then she would get antibiotics, and they didn't really work for her. Other people would get cured, and for her, it didn't quite work. And again, why is that? So tonight we'll answer all of those questions. So you're going to feel very productive by the end of the night. So before we are going to talk about examples of all the different classes of drugs we have to fight possible invaders that might harm our health, it's helpful to think of what are the possible things that can invade us and cause us harm. And if we're thinking of all the microorganisms that can sort of start living in us and causing us harm, we can generally classify them in 5 different categories. The first one is bacteria. Those are what we call prokaryotes, so their life form is slightly different than ours, but we have many, many things in common as well. For instance, they don't have a nucleus where they keep their DNA, and when we start talking about the drugs, we'll look at more differences between us and bacteria. But these are the things that can cause pneumonia, or staph, the little thing you saw swimming around, is one of the bacterium. The second class of drugs, organisms that can cause us trouble, are fungi. And these actually are closer to us than bacteria. Fungi are eukaryotes. They have a nucleus just like us. They make proteins just like us. And this is important in trying to develop drugs for these organisms, because as we will see as we march through all of these organisms, what we want to do is we want to kill the thing that is invading us, but we ideally don't want to touch any system in the host or the human being. And in order for us to design drugs that kill the bacteria but not us, we need to find differences between us and whatever it is that's invading us. And because fungi are eukaryotes, and we are eukaryotes as well, it's much harder to find drugs that can target fungi. So we have millions exaggerating, of course, but many, many, many different drugs against bacteria. We only have, let's say, 6 classes of drugs against fungi, and that's because they are so similar to us. The third class of microorganisms that can cause trouble for us are protozoa. Those are single-cell organisms, and they're divided in subclasses based on how they move. And one of a sort of infamous example of a protozoa that causes mankind lots and lots of suffering and death is the malaria form. And so especially in nations such as Africa, many children die of malaria each year. So this organism is referred to as man's biggest killer. I mean, so these organisms are highly impactful. And again, if we don't have drugs, they can cause a lot of harm. The fourth class, helminths. Helminths is a fancy word for worms. And this infection is widespread both in the Western world and in less developed countries. Very common in, let's say, daycare, childcare centers, as well as other places where people are together for a long period of time, sometimes mental institutions or daycare situations, whether it's the very old or the very young. Often there are outbreaks of worm infections that we can treat very easily with drugs. And then the last group are viruses. And viruses, again, are very different from bacteria in the sense that they are so clever. They don't have a lot of machinery to make proteins, to make more of themselves. They are so clever that they insert themselves into the host. They hijack all our equipment, so they take our stuff to make more proteins. They take our machinery to make more DNA. And then once they're done, they exit the cell again. And we have done all the work for the virus. And the virus didn't need to spend any energy making more of offspring viruses. And this, again, implicates that it's really hard to find a target that is specific for the virus and not the same as our target, because the virus actually uses our machinery. So if you make a drug against that machinery, automatically you're also going to affect the host. So again, it used to be pretty hard to have specific drugs to treat viruses, and an enormous amount of research has gone on to develop many, many more drugs, especially stimulated by the HIV epidemic. And we'll talk more about that next week. But what all these organisms sort of unite, or our unifying approach to trying to kill or stop the growth of these organisms and not host us, is we are going to find what's different between the thing that's invading us and our human cells, and we're going with drugs target that particular difference in order to kill the bacteria and keep ourselves healthy. All right, so what are some examples of drug targets? The first class, very famous and very, very useful drugs, are in fact penicillins. You may have heard of the drug penicillins. And we'll talk about these drugs target the cell wall, which is an extra wall that bacteria have around themselves, and we don't have that wall. So if you think of a human being as a house, we just have a single wall. If you think of a bacteria as a house, but a much smaller house, it's insulated. It has a wall, and then it has an extra wall on the outside. And so by as we'll see in a minute, the penicillins are going to break holes in that wall, and the bacteria will be leaky. And because it's unique for the bacteria will die, and we're fine because we don't have that wall in the first place. So if you break it, it doesn't do anything to us. Secondly, we have an enormously efficacious or powerful or strong group of drugs that interfere with the ability of the bacteria to make new proteins. And proteins are things we need, and the bacteria needs it as well to live a life and be functional. And You heard last week that some of the things we can replicate, some diseases we can replicate in mice or even reptiles, and the process of having DNA, making RNA, and then making that into protein, so the actual stuff of life, is very similar in bacteria and in us. But the actual machinery, how you make the coding of the template into a protein, is different enough between bacteria and humans that we can target the machinery in the bacteria and not so much inhibit the protein synthesis in humans. So we'll go through a couple of examples of these drugs because you will recognize maybe some of the drugs as they're widely used for common conditions. And then next week, we'll start talking about interfering with differences that bacteria and we have in how we handle DNA synthesis, which you need to make more of yourself. And then also specifically for fungal drugs, I mentioned that fungi are eukaryotes, so they have a cell. They look more like us than the bacteria do. And one of the main differences between fungi and us is that we have in our cell membranes cholesterol. We all hear about cholesterol. Cholesterol is bad. Well, actually, it's not that bad because we need it for our cell membranes. It's too much cholesterol that's bad. But cholesterol is very essential for us to maintain our cells. And fungi have something that looks like cholesterol. It's called ergosterol, but it's different enough that we can have drugs that all target the cell in the fungus, the ergosterol, which is the fungus form of cholesterol. And because we don't have it, again, the drugs will target that specifically. So again, we're not going to walk out of here and ask you to list all these, but just an example of we have many, many different ways in which we can target those things in the bacteria or the fungus or the virus that is different from us and that will attack the organism. So before we get into the details of the drugs, if you kind of think of the last time you got your doctor prescribed you antimicrobials. How did your doctor even know which one to pick? Because I just told you we have hundreds and hundreds of antimicrobial drugs, and they tend to give you one or two, right? And so how that works is the doctor will listen to, what your history, like where you've been and what your symptoms are, and your doctor will know, or the hospital will have found out about susceptibility of organisms that are sort of living in the community around here. So as, let's say UCSF, lots of patients come here, anytime there is an infection, the organism will be isolated and will be grown in the lab, and it will be tested against all, let's say, all known antibiotics to men. And then we will look what the susceptibility of these bacteria is to all these drugs. And so how that is done is we are defining what's called the minimum inhibitory concentration, or MIC. And this is a lot of words, so let's just look at a picture. How that works is you see here a petri dish, and it's sort of yellowish, and that yellowish is lots and lots of bacteria. And to not confuse you, I'm sticking to the same one all the time. This is Staph aureus, so the same little guy that was trying to escape from the neutrophil but didn't make it. Well, this plate is covered in Staph aureus. And then these black dots are little discs with drug in it. So sort of like a little piece of tissue. And then let's say this piece of tissue had 10 drops of drug. Then this drop had 100, this had 1,000, 10,000, 100,000, and 1,000,000. So we go more drug, and then we see, you can clearly see here, around this little disc, there's a very large clear area. So the drug is sort of seeping out of this little piece of paper, and wherever the drug is, the bacteria can't grow. So we can see this drug is working against this particular organism. And so we can sort of count, if we only give 10 drops, it's not going to work. But if we give 1,000, well, maybe there's a little bit of work. And if we give 10,000, so you can determine a concentration at which we are going to see inhibition of growth. And that way we determine if we can use this drug against this organism. And so each hospital has enormous databases with this data showing that, for instance, all the E. coli that lives in the community, so in you and in me and on you and on me, and when all the, let's say there's a lot of patients that come in with urinary tract infections, the doctors go into the computer and say, at UCSF, what's the best drug to pick, because maybe if there's local resistance, it's not useful to pick a drug that's not going to work. So this information about which drug will work for which organism is constantly accumulated and updated as we speak, like the labs are running right now, and we update those data and put out new reports yearly. So that's susceptibility. Will the bacterium be killed by the drug? And so let's see what happens if we sort of look in more detail. If we give a drug, how is that working? Well, here we have these green balls are, let's say, E. coli, and these organisms are very quick at changing genetic information. So they tend to be, similar, but not all the same. So these guys are all E. coli, the green ones and the red ones. The green ones grow very, very fast, and they are all competing for food. So if you grow fast, there's only so much food, so the green ones start to win out, and the red one is a little slow, maybe because he is spending a lot of energy holding on to all sorts of mechanisms to become resistant to certain drugs. Cost extra energy, can't grow that fast. So now we're going to give drug A against E. coli. Spare you the names for now. And lo and behold, lots of bacteria are dead, so all these green ones are dead. And we have only a few bacteria left. And actually using drugs is a numbers game, because at this point what happens is our defense is going to kill the remaining cells. And this is key. And in most of us, because we're all here, we didn't die from the infection that we got our treatment for way back when. So we're all happy patients. But now, let's think back to Elizabeth, right? So now we know, okay, we have a lot of bacteria. We take a drug. It reduces the numbers to manageable numbers that our immune system can deal with. But what if you're Elizabeth, right? Sometimes she would get drugs, the numbers would go down, but not far enough that she could deal with it. So even if she would receive drugs, she may not always have gotten the numbers low enough that her limited ability to respond was able to finish the job. And so in real life, in those cases, sometimes they need treatment for even longer than the standard regimen that you and I would receive, or they may need higher doses. So it's an important concept to keep in mind that actually when you take drugs, you're doing part of the job yourself as well. So good job, everybody. So that's one thing. So, okay, so we answered question 4. Why was Elizabeth sometimes not helped by antibiotics? So let's then go backwards and say, why do you, and me too, always have to finish a prescription when you are given one? So let's go back to our blob of bacteria. Let's say you have upper respiratory tract infection, you're coughing, you've been to the doctor. They said, yeah, you have upper respiratory tract infection, you need antibiotics. So you start taking your antibiotics. Let's say you get a Zithro pack, and we'll talk about what that is later. And so you are taking it after 4 days you feel great, so, I don't like taking pills, I'm gonna stop. But what happened is, after 4 days, you've killed off all the green guys. And all the red guys, maybe they were inherently resistant. If you keep on taking the drug, it may just take a little longer for the red guys to die. If you keep on taking the drug, eventually there's not that many red guys left, and your immune system will do the job. If you stop too soon, you have shown the red guys what the drug looks like. They have been able to live in the presence of the drug, so they had time to figure out a way to do something to the drug or do something to themselves that allowed them to not die. And bacteria are very quick in giving each other a little bit of genetic information that can carry resistance. And it's sort of like kids with baseball cards. It's like, oh, what do you got? Oh, whoop, here, boom. And this can be happening in a period of 24 hours. So they've done studies where nobody was resistant, then a patient with a resistant E. coli came on the floor, and within 24 hours, suddenly the E. coli in the other patients was resistant as well. So this can happen very, very quickly. And so if you or anybody gets antibiotics, you stop too soon, you are now you have selected out, all the weak ones are gone, but the kind of slightly more hardy ones are left. If you stop taking the antibiotic, these guys are now going to grow out, and you have now created or selected, as it's referred to, a resistant population of bacteria. And then you may go around and spread this to other people. So it's really important that even though you feel better, you always have to finish your prescription in order to prevent spreading resistance to other people. All right. So back to the doctor's office. So we said you come to the doctor, there's so many drugs, I was going to say a bad word. How do they know which one to give? So first of all, they need to match the correct drug to what they think you have, but they also need to take into consideration what's going on with the patient. Because I'm going to go here first, some drugs are going to, for instance, if we start at the bottom, some drugs may be very good at killing bacteria, but they might also be very good at inducing damage in an unborn child. So if a person is pregnant, you may not be able to choose from any drug you might be able to choose from if, let's say, your patient is male. So that's something your doctor would consider. The patient age. There are some drugs that we'll talk about next week that are contraindicated in children who are still forming their bones. The drugs interfere with calcium. You don't want to interfere with bone growth in young, healthy kids. So some drugs are contraindicated in young children. Some patients might have a true allergy to, let's say, penicillin. If your patient is allergic, of course your doctor should not and will not prescribe penicillin. And then this is an interesting too, how severe is the illness? Because some cases, if you have a little bit of, let's say fungal toe or sort of a gym, whatever, fungal toenail, it's probably not going to kill you within the next 2 days. So you can sort of try this. If it doesn't work, you can try something else. But if your patient has life-threatening meningitis, it's probably really important to get it right and to go in with the most aggressive treatment you can possibly think of, even if there is a risk of, let's say, kidney damage. So it's always a balance of risk versus benefit. But for the most part, as you probably all have experienced, the drugs that we have to treat antimicrobials are very well tolerated. So, of course, we look at what's going on in the patient. And then this is an interesting differential, too. When we look at drugs, we can put them in two buckets. One we call bactericidal, and cidal means to kill. So when you put that drug on top of the bacterium, it will actively kill the bacterium. It will kill everybody They like to say, kill 'em dead. So that's the end of that. Bacteriostatic means it will stop the bacteria from growing, but the guys that are already there are going to live as long as they would normally have lived. So it will take you slightly longer to get rid of that infection, but sometimes that will still work perfectly fine, and you only want to worry about, do I really need to kill them that fastly when your patient is having this severe life-threatening meningitis, inflammation of the brain and the spinal cord. So these are all considerations that go into the equation when your doctor picks an antibacterial drug to help you or to help your immune system finish off the job. Yes. The question is there ever a situation in which you would use both classes of antibiotics? And in fact, one example would be when there is meningitis, so again, inflammation of the brain. If it's viral, often it may resolve on its own, but if it's bacterial, we really need to treat it fast and aggressively. And then we use a drug, actually one of the penicillins that we'll talk about in a moment, to break open the bacteria, and then we use another drug to now enter the bacteria that we just broke open to kill it even further. All right, so we're getting to the drugs now that you've been waiting for. And here we see Sir Alexander Fleming, who actually turned out he was sloppy one night. He was working in the lab. He was having a petri dish with Staph aureus again, the highlight of the night. And he left it out, and he came back a couple of days later, and he noticed that not only did he have this plate full of bacteria, but then mold grew on top of it. You know what you get when you leave your coffee filter and you don't, that sort of stuff. So he had that, and first he thought, oh, what a mess. Then he looked closer, and then he saw what we just saw on that petri dish. Around the mold, the bacteria couldn't grow, and in fact, the bacteria had all died. And so he was clever to make that connection, thinking, this mold is producing something that is killing bacteria. This could be extremely useful as a drug. And so he really was the first one who discovered that. And then his two colleagues found out the chemical way how to isolate penicillin from the mold. And the three of them then eventually received the Nobel Prize for the first antibiotic that has been clinically so useful, and in fact to date still is. And so here is a nice humble quote from him. And so, one more time, penicillin, this is the prototype of the class of drugs that interfere with the cell wall. And just to show it one more time, here it looks like a peanut, but it is a bacteria. And the DNA, the genetic material, is just floating around in the inside. And the inside of a cell we call the cytoplasm. And then you can see several layers. There's this sort of gray layer, which is the membrane, the outside of the cell. And then the bacteria has this whole thick wall around it to protect it from the outside world. And the drugs we're going to talk about, and penicillin in particular, are going to attack this wall. And we'll get to these other names later. So for now, just think about penicillin is what we're talking about. And so again, we have, just to remind you, this is our bacterium. And if we're saying this is the inside of the bacterium and this is the outside, and if we zoom in on the wall, we can see that it looks almost like those doormats with those sort of, octagonal shapes. Or if you think of knitting, it's like a quite intricate structure that is pretty hard. So think of a hard plastic mat. And if we look even closer, you can see that there are purple and blue structures. And these are sugars. And these sugars are going to be connected with a cross-linking peptide. Now you don't necessarily want to worry about how that goes, but you can imagine if you're trying to make a really rough wall and you make these big sheets and then you connect all the sheets both horizontally and vertically, you can get a pretty sturdy structure. And so the cell wall is really these sheets of sugars, and then they are hooked together, and so you really get this pattern of a doormat. And so the bacteria spends quite a lot of effort making this strong wall, and if you have sheets that flow across each other, it's not rigid. So it's key that all these sheets are linked together, and once they're linked, they're never going to come apart unless something bad happens to the bacteria, and that bad that is going to happen is penicillin. So the word for the little machine, if you will, that is making these really hard cell walls for the bacteria is called a transpeptidase. It's the little enzyme that links all these sheets together. And what happens is that penicillin likes to bind to this enzyme and prevents it from doing its job. So now the bacteria is trying to make a wall, it's trying to lay the bricks and put cement in between, but penicillin is preventing these sheets from being stuck together. So everything is just floating loose and it's not forming a wall. And let's see that in a second. I thought I had another slide, but this is fine. And so, again, the penicillin, what it does, it binds to this little enzyme, the transpeptidase, and because of penicillin being so efficacious, they changed the name of this enzyme, whose function it is really to build the wall for the bacteria, and they changed it and they named it penicillin binding protein, you know, the target for penicillin. But of course, it's really the bacteria had it all along to make a wall, to keep the outside out and keep its insides in. And so it inhibits this cross-link, and if you inhibit this cross-link, the bacteria is leaky, and what we'll see in a movie pretty soon is the bacteria will die and just burst open and pop. And we'll get to this in a second. So let's look at that. So you see these things are E. coli, and they're growing. You see this guy's longer and longer, and then we add penicillin to it, and they're going just pop. We don't have sound, the popping sound, but as soon as the penicillin is added, they are all going to, boom, there it goes. And if we waited there, another one, boom, 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 boom. So it works, right? So if your neutrophils are not getting the bacteria, clearly the drugs will. And so truly these drugs, they poke holes and the E. coli explodes. So pretty useful. And many of us have probably experienced penicillin in action. And so if you are excited by chemistry, this is your chance. If you're not that excited, don't worry about it. But what is interesting about the chemistry of penicillins is that if you look at this structure here, you can see that the structure, this is the chemical structure of penicillin, and you see that there is a ring that is square. And in nature, not many things are square, and this is a pretty unique structure that is very typical for penicillins. And this square form plus this sort of house-shaped form together is what's referred to as a beta-lactam. And the bacteria, of course, are thinking, we don't like being poked and then blown up into pieces, so the bacteria are trying to come up with a clever way to not be sensitive to these penicillins. And what the bacteria have discovered is they can make little Pac-Mans that really specifically go in and chew up this square structure, chew up the beta-lactam. And so these things, little Pac-Mans, just come out and chew up the penicillin, and then the bacteria lives and the penicillin is no longer active. And so this is called a beta-lactamase. It refers to the fact that it is an enzyme that eats beta-lactams, and beta-lactams refers to the chemical structure of penicillin, and this, especially this square part, is very unique. Question?
I have a question.
Yes.
I've always wondered how close to that schematic sort of
Display.
configuration is it really?
Is
I mean,
s-
what
when-
do you see?
If you look with a microscope, because you can't see it with your bare eye, but if you look with a microscope and you were to sort of visualize this structure, it is pretty rigid, and it's really, those four atoms are really sitting in a square. It is really, I mean, you'd have to sort of imagine there are different ways of visualizing structural molecules. Sometimes it's sort of little bulbs and each atom has a different color. But the configuration of these penicillin or beta-lactam structures really sit in a square, which is very unusual in nature. So if you look at it, whether it's electron microscopy or a model form, you will recognize that square, which is very unusual.
What about like double bonds? Do they look different, or do
It
can you
depends
see
on
the difference
how.
between hydrogen and oxygen
So it depends on how you visualize it, because you know the chemical structure and then you color code back in. You make a 3D model and you give different atoms different colors. So then you can see it, but you can only see it after you've colored it back in. All right, so bacteria are clever. They don't want to be killed by penicillins, and they create these little Pac-Mans that chew up the penicillins, and now we need to come up with the next generation of drugs to still be able to keep all of us from walking around with infections the whole time. Question. The question was, are the carbon atoms in this structure different, and is that why they sit in a square? It's the combination of having the nitrogen, the double bonded oxygen, and these side groups. So it's the very, it's not that there are unique carbon structures. This structure in and of itself is unique. One more and then we'll move on. So the question is there a carbon at each square? And so when there's a ring, each time there is a kink, for instance, here is a carbon, here's a carbon, here's a carbon, and we draw the ring around it. Thank you. So the Pac-Mans is one of the main ways by which bacteria fight back and chew down the penicillins. There are other ways by which bacteria can become resistant to penicillins, and one is by changing the very thing that the penicillins bind to, or preventing access of the drug into the bacteria. So, like I said, you know, the hospitals generate these data based on the things that grow out of all of us and the patients. And so when are these penicillins used? Because of their structure and the nature of the bacteria, there are certain combinations that are very effective and certain bacteria that are just not sensitive to penicillins. And so skin and soft tissue infections are very, very susceptible to penicillins. You heard last week that our skin is covered in bacteria, especially staph. So staph epidermidis, so staph that lives on the dermis, staph that lives on your skin, is very susceptible to penicillins. And many, I don't know, but maybe you have had elective surgery, and then when you go home, so the physicians break your skin, there's a risk that the bacteria that live on your skin go inside. Often you get sent home with Keflex or a first-generation cephalosporin, and that's a drug that works just like penicillin, killing all those bacteria dead, So there's only a few left for your immune system to finish off. Throat and skin infections, so people who've had strep throat, often the drug of choice to use would be a penicillin. And then meningitis only if it is non-viral. So meningitis caused by Neisseria meningitidis or strep pneumo may respond very well to penicillins. And in that case, the scenario would happen as we talked with the gentleman earlier, that you may use multiple drugs to really make sure you're treating the patient aggressively. So to this day, penicillin is still used, which is pretty remarkable given how long it's been around. So you can see here, 1942, this was the very first patient that was cured by penicillin, and until that time, anybody who would get this infection, either gonorrhea or syphilis, was very bad off. And to date, penicillin is still the drug of choice, so the treatment to treat syphilis. So if I say it's so great, I've shown you a movie that the bacteria are killed, what's the problem? Well, there are some problems. So some patients, if you need treatment with high doses of antimicrobials or antibacterials, that's the same, means the same thing, you can suppress the growth of the bacteria that we normally have in our GI system. And so if you suppress the growth of the bacteria that are normally in your gut, some that are more resilient can now start eating all the food that was normally shared by everybody in the gut. And these guys can outgrow. And then in some cases, that can lead to what's called bacterial colitis or pseudomembranous colitis, caused by the agent or the bacterium Clostridium difficile. And that can be a very, very painful, very strong diarrhea. And even though it's caused by a use of antibiotics, the treatment is a different antibiotic, which then kills C. difficile, and then everybody's happy again. So because we normally have flora or bacteria in the gut, anytime you use, especially the higher you go and the longer you need antibiotics, some of the bacteria in your gut are being killed by the antibiotics. The bad guys can outgrow, and that's why many people can get diarrhea. So this can happen with any of the antimicrobials. In addition, I'm sure you have heard about this, that a small or a portion of the population is truly allergic to penicillin. And when we say truly allergic, it means that when they are exposed to penicillin, within 72 hours, so relatively fast, they experience rash, swelling, itching, which can progress to true anaphylaxis. And true anaphylaxis means a very severe allergic reaction where all your blood vessels dilate, you don't have enough pressure in your system to keep blood pumping around almost, and you get your immune system is going nuts and everything swells up, including your windpipe. And if you're not brought to an emergency room, your swelling will prevent you from breathing, and that may be lethal. So it's a very, very serious condition, and that's why people who are truly allergic have a little card, and the doctor always asks you, are you allergic to any medication? Because we know people can be truly allergic to penicillins. Question? Oh, thank you for asking. So if you have this rash, you are not truly allergic to aspirin. So people who have been exposed to ampicillin, so there are many, many generations and many flavors of penicillins, and quite a lot of people actually get this particular rash when they are exposed to ampicillin. This is not the same as a true allergy where you would progress to life-threatening swelling of the windpipe. And many people, though, you know, when the doctor asks you, are you allergic, and you think, well, I had a rash, you're inclination is to say, yes, I'm allergic, but you may have just had the ampicillin rash, which is not always associated with a true allergy. So the question is, if you have ampicillin and you have this rash and you continue taking it, will it progress to anaphylaxis? If you're not allergic, it will not. If you had the rash and you worry about it, they can do tests to determine if you're truly allergic or if you are sensitive to ampicillin, but you are not at risk of progressing to this windpipe swelling and all the associated risks. Excellent. Rash is not life-threatening. So, as we said, or as I said, so we have penicillin, that was the first one, and it really was a miracle. So many lives are saved. And then the bacteria started fighting back. They made those little Pac-Mans chewing up the penicillins, and they changed a little bit the nature of how they make their walls, so then it was harder for the drug to attack that. And so we didn't want people to keep being infected, so then we needed newer drugs that could still kill the bacteria that are now no longer sensitive to penicillin. And again, here you see that unique square structure that is characteristic of the penicillin family. And here is even a larger close-up of the bacterial cell wall, where you see before we had purple and blue, and then the little chains that are cross-linked. Now, just because it comes from a different book, they're purple and green. But again, you can see the peptides and how they are going to be cross-linked together. And so the penicillins are preventing this cross-link from happening. And we saw that's very effective. Boom, 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 E. coli explodes. And for the drugs that are resistant, we have a wonderful different drug that is binding not to the same cross-linking enzyme, but actually just starts hanging on the cell wall. And so this drug is saying, oh, I see this chain of peptides. I'm going to sit right on it. And now by sitting on it, it's sort of like defense in basketball. Now this red link that needs to cross-link can no longer reach its target because this drug is just sitting in the way. It's truly just being a big old bulky thing that gets in the way of everything. And so now, again, we cannot cross-link the sheets together. The bacteria will explode. And so why is this so important? Why are we so excited about something that is like penicillin, but a little bit different? Well, I'm going to show you why. And this is again going back to our star of the evening, Staph aureus. Staph aureus, when we had penicillin, it was invented in the 1940s. We were treating everybody. It was great. But then after about 10 years of using it in the community, almost everybody was resistant to penicillin. So now we didn't have anything to treat our patients with. So then we said, okay, bacteria fight back. We can come up with a different drug that is going to kill you. So then the drug companies developed Methicillin, and this was a slightly different flavor of penicillin, and so now we could again kill all the bacteria. That lasted for 10 years or so, and then the Staph aureus was like, I'm clever too, so then it became resistant to methicillin, and that became the infamous methicillin-resistant MRSA, or the hospital sort of super bug that for a while we were worried because we didn't have any more drugs to treat the methicillin-resistant staph. But that's where the clever people came. What if we try to hit the bacteria through this different way? What if we use this guard that doesn't need the exact same mechanism? So that was vancomycin, and that was really, again, a revolution. Now we had a good drug that could kill the bacteria that were resistant to all these forms of penicillin. So that was really great, and that lasted quite a long time, but now this is where we kind of get into the downer part of the talk. Then in 2002, even vancomycin-resistant bacteria were discovered, and at that time we didn't really have anything as a backup. So we're like, okay, we've run out of options, and so that was pretty scary because people were now getting infections, getting really resistant infections, and we didn't have anything. Of course, now we do, thank God, have many options to treat vancomycin-resistant organisms, but it's definitely, you can see the progression that we provide a drug, then the bacteria become resistant, then we have to come back with a different drug, and this is sort of a tug of war ongoing, and because it can be really hard to reach a point when we no longer have a treatment for somebody who's very infected. That's why it's so important that we all have to play a role in not, taking antibiotics when we shouldn't take them, because we shouldn't be out there making bugs more resistant than they already are, because we really have a joint responsibility in that sense. Question. So the question is, we're getting so clever at using bacteria, and so we must find ways to use bacteria to find methods again, or yes, but remember, because penicillin in the first place is a natural product. It was the mold who made it against the bacteria. And so it remains an ongoing battle. But thank you for asking that question. Like, we must be so clever, we must be able to do it. What's at play here is also that the drugs that we have on the market today are being brought to market by pharmaceutical companies. And pharmaceutical companies want to help people, but they also want to make money. And if you think about drugs that can make money, that are drugs that are going to, somebody's going to need them for the rest of their life. Hypertension, lifelong drugs. Anxiety, probably not cured within a week, so you can keep selling that drug for a long time. If you make a good antibiotic drug, the patient takes it 10 days, and they're done. So you're only selling your drug for 10 days. So there was a little bit of an interesting lack of enthusiasm by the pharmaceutical industry to make drugs that were maybe, the better your drug, the shorter your patient is going to use it and the less money you make. So there wasn't necessarily the biggest financial incentive by the pharmaceutical industry to make these drugs. And to make this point, so this slide shows the number of patent applications submitted to the regulatory offices of the FDA by pharmaceutical companies. And so you can see lots for depression, anxiety, bladder function, maybe that has to do something with, I'm not going to say that, osteoporosis, antibacterials, right? It's in the bottom top three. So there was a financial lack of financial incentive. Question. So the question is, if everybody has a different immune system, why prescribe an antibiotic for 10 days or 14 days? Because isn't the last part overprescribing which causes resistance? And it's the underprescribing that causes resistance when half of the bacteria are dead and the remaining bacteria are still too many in number to be killed by your immune system. And they now have the chance to keep living. Whereas if you kill everybody and there's only 2 left and your immune system can take care of them, and then you take pills for 2 more days because maybe you're very effective at clearing the bacteria, there are no bacteria in your system that you are making resistant. So too little is the problem. 1 or 2 more days in your particular system versus mine is not the problem. So the question is, what about overprescription or people who take antibiotics when they don't need to? And so, for instance, you can buy stuff on the internet just for who knows what. And in fact, that was a big problem when there was the anthrax scare in Washington, and everybody was thinking, oh my God, I have to be prepared. And one of the drugs that is the drug of choice for anthrax is ciprofloxacin. And so everybody started buying ciprofloxacin from Canada, and the hospitals, you know, in the neighborhood, like normally, were collecting data on resistance. And within a couple of months, everything in the surrounding of the Washington area was resistant. So if people were taking it just in case, like I'm just gonna take it for a couple of days. And so that immediately led to resistance. So the question is, I've read that a lot of the antibiotics are fed, or 80% of the antibiotics that are used are fed to the animals that are used for food production, and what's the relationship to us. And so for some of the antibiotics, it depends. One part is that a lot of it has to do with organisms that live in a GI system anyway, so sort of like worm prevention, lintworm, heartworm. Those drugs tend to stay in the GI tract, so they kind of stay in the food system and exit in the food system and don't get into the meat, which is good as if you're eating meat. The excessive exposure, though, definitely has sped up development of resistance of those organisms that were in the gut. All right, and just given that we're talking about this system, again, Staph aureus is the highlight of the night, and just to wake you up a little bit, it is a problem if you have this infection, and if we run out of drugs, it lives on the skin, and it can, you know, it will start burrowing into the skin, and the people will have ulcers and ulcers and ulcers. And if we don't have any drugs to treat it, it is a miserable way to be. And so anatomically, this would be the last part of your GI tract. Just to orient you all. All right, so far, you have sat through and survived, and even were very interested in the chemistry of the first target that we have in all the classes of antimicrobial drugs. So we're not going to go over every single one of them, but this was really a key starter, and congratulations on making it so far. Are we still awake? All right, so now let's look at the second class of drugs, and these are acting by inhibiting protein synthesis. And so we all make proteins every day. We do, bacteria do, fungi do, everybody does it. And we're just going to, again, sort of paint a very broad picture of what's going on in a cell. And because we like to talk about ourselves, I have a eukaryotic cell, meaning a cell of a higher organism such as ourselves. And what you see here is not an Easter egg, but it's a diagram of a cell. And the blue thing is the nucleus. And in the nucleus is this purple squiggly, which is our DNA, where we have all our genetic material. And when we need to make proteins that will be able to execute functions, first what we do is we transcribe the information on the DNA into what's called RNA. And then in humans, we kind of modify the RNA so that it's safe to exit the nucleus and go into the cytosol. And then in the cytosol, this RNA is now our template that has written on it, make this amino acid, make this amino acid, make this amino acid. So it's really like a line of information that is going to tell you going to tell the machinery in the cytosol which parts it needs to add onto the growing chain of the peptide. And you can see here these little, I don't even know what to call this shape, but let's say there's two parts to this machinery, which is called a ribosome. And these two parts come together and they clamp down on this RNA and they start reading the RNA. And as this machinery is sort of hopping along the RNA, as it's doing it, it is churning out a protein molecule. So you kind of can think of it as crocheting. You know, you have a structure, and then you do something on the top, and then on the bottom, this long thing of your product is coming out. So that's kind of how you want to think of these ribosomes making protein. And it's these little busybodies, so sort of the crocheting machinery, that we are now going to target with these drugs. Because you can imagine, if you inhibit this bacteria from making any proteins, it can't make an enzyme, it can't make a Pac-Man, it can't make a piece of membrane, it can't make anything it needs, so eventually it will die. And so there's steps involved, and when you think about understanding all the steps that are happening in the process of moving from DNA to proteins, we could target the different steps with drugs and see which bacteria are going to be most sensitive to this. The size of these molecules of the crochet apparatus, if you will, are very different between bacteria and the human form of the crocheting machine. So we can have drugs that bind to the bacterial crocheting machine, but actually leave ours fine, so we can kill the bacteria, but we can just stay fine and dandy. Because we always want to target the difference, keep our own machinery just fine. Eye on the prize. And again, discovery of some of these compounds that are able to interfere with protein synthesis in the bacteria was done in this case by Dr. Waxman, and again he received a Nobel Prize for his work because the protein synthesis inhibitor that he discovered turned out to be very effective in the treatment of tuberculosis. And you may have read about TB. You know, in the old days everybody was dying and was put in those sanatoria where they were sort of aired and sitting outside, but still everybody was dying until they found these drugs that could cure people from TB. So interesting, so we see how these drugs work. They target the ability of the organism to make proteins, and then we have different flavors. So one class is called aminoglycosides. Most likely, if you think of your own experience going to the doctor getting some pills, then you will not have had these drugs because they are used currently in the hospital intravenously. So they're very strong drugs, if you will, and are used for very severe infections. And you can see their names are all very similar. They end in mycin, streptomycin, gentamicin, tobramycin, amikacin, that's different. And neomycin, you may have seen, this drug is so toxic because it's so good at inhibiting protein synthesis that we don't want to put it in anybody's body, but you may have seen it if you go to your pharmacy in the triple ointment. Neomycin is the component that is in there as one of the active ingredients. And so I'll get you in a second. And so now you know how it works. It inhibits protein synthesis in the bacteria. Question? Very good question. So the question is, we were just talking about cell walls that are so hard. Now how do these drugs get to the inside? So all these questions come together beautifully because the example earlier of how are we going to treat somebody with meningitis, in fact we give them a penicillin to break open the cell wall, then we're going to add an aminoglycoside to really make sure everybody's dead. So the wall's already destroyed in a combination therapy. And secondly, there is a big old wall, but then the bacteria, just like us, makes metabolic byproducts and needs to get food in. So there are special channels that allow stuff in and that also let waste out. So the drugs have to go through those channels to get into the bacteria. But when we are really worried, like with meningitis, these guys are always given with a cell wall inhibitor. Very good question. question in the end. So the question is, tuberculosis has been in the news lately a lot, and again, it's becoming resistant to many drugs, and how come we use, I'm saying here, we can use aminoglycosides. So TB, we can talk another 2 hours about TB by itself, but there is a standard regimen which is rifampin, isoniazid, pyrazinamide, and ethambutol. So four drugs, because we are so worried, so 4 drugs at the same time. And this is now more one of the backup drugs. And so the extremely resistant form is resistant to many, many things, and that's why we're going back to other drugs and older drugs. So this is historically, but we now have different and better drugs to use. But we can talk more about the XD TB afterwards. Question. So the question is, how do we get drug-resistant organisms when there's so many drugs to choose from?
Yeah, like a location where they're like, sorry, you tried everything.
Right. So even though we have many, many drugs, not all drugs work for all organisms, and tuberculosis in and of itself is one of the more difficult organisms to treat. It's a mycobacterium. It's really small, and it lives inside cells. So first you have to get to it, and then because it grows so slow, it is really hard to treat, because the drug can hang around for a long, long time. There's a lot of opportunity for resistance. And the 4 drugs that we have really are the main drugs for TB. So we have many, but for TB, really, we only have 6 that are really, really effective for TB, for example. One more, and then we'll move on. Yes? So the question is there research going into inhibiting the transport of the mRNA out of the nucleus? And so this is where I have to say, I'm sorry I showed the eukaryote, because the bacteria doesn't have a nucleus, it's a prokaryote. So that would inhibit what's going on in us and not in them. So yes, there's research, but not to try and develop antimicrobial drugs. But there are actually people doing research on transport in and out of nucleus, which is a whole different story. But it's a very good thought. All right. So moving on to another, within this category, there are many, many drugs. And another one is macrolides. And you may have heard of this drug, or maybe some of you have even taken it, because this drug is very, very good in treating respiratory tract infections. So again, this is a drug that targets the crocheting machine in the bacteria. And in particular, it's also good at Bordetella. And you may know that a couple years back, Bordetella pertussis causes whooping cough. And every now and then there's outbreaks of whooping cough in the schools and in the area here. So I think it was 2 years ago or something, there was quite a significant outbreak of whooping cough. And then, thank God, we have the drug, the macrolides, and we can suppress this from going around, and patients from getting very, very sick and getting lung damage. So for those of you who have taken these drugs, the trade name is Zithropac or Zithromax, and often it's a pack with just 5 pills, and you take 2 the first day, and then just 4 more days, and the drug stays in your system for 2 weeks. So you take pills for 5 days, but you're actually being treated for 2 full weeks. And it's very, very good for many of these organisms that give you respiratory tract infections. And last week, Elizabeth mentioned using this drug as well. So respiratory tract infections, you use one of these drugs that inhibits the crocheting machine. Adverse drug reactions, GI upset. Now you guys are pros. You know, anytime you take an antibiotic, you may disturb the flora that normally live in your gut. You're going to have an imbalance, and you may have diarrhea. So now, anytime you see that, you guys know that already. And the last, question.
Before that,
yes.
You did go over the adverse reactions for the
right.
glycosides.
because I thought maybe that would be too boring and you wanted to hear about whooping cough, but I'm happy to. So aminoglycosides are interesting. Their structure makes it so they're very, very water soluble. And what that means is when you take this drug, they don't want to hang around in most of your body that has a lot of cholesterol and lipids. They really like to go into places where there's a lot of water. So that would be your bladder. The way you get rid of those drugs is you would pee them out, everything that has to do with water. And so because so much of this drug really concentrates in your kidneys and in your bladder, we see that these drugs can be nephrotoxic. So they are toxic to the kidney. And when you're in a hospital here and you have such a severe infection that you need these drugs, nephrologists will come by every so many hours and check your labs to make sure your kidneys are doing okay. It's that toxic. People come by and check your kidneys just to make sure we're not damaging your kidneys with these drugs. And in your ear, we all have, we can all hear and we can maintain balance, and there is fluid in little tubes involved in that. So again, the drug will go to the fluid because it doesn't like to hang around in the cholesterol, and it concentrates in the ear in those fluid portions, and so it can cause ototoxicity and vestibular dysfunction, meaning that the patients are no longer able to keep their balance and they might actually get dizzy. And if you have an interest in histories of litigation around drugs, there are people who had been exposed to these drugs at relatively high concentrations. Then they were cured from their infection, but they were not able to keep their balance. And that's, if it's going on for a long time, that can be irreversible. Question?
So it can affect our ribosomes? Is it the same?
It's a different mechanism. It's not through ribosomal inhibition, but good point. I'm going to talk after about that. And in part because I'm not sure I know all the details. And so here we were still in the crochet machinery inhibiting drugs, talking about these drugs that are really, really good for respiratory tract infections. And again, we have our expected GI upset. And the last class of the night, so we can relax and we're almost there, are tetracyclines. But again, these drugs are relevant, and maybe not to put the high school students on the spot, but these drugs are very interested and very, very efficacious in treating acne. And so acne is not caused because you're not clean, because you're not washing. It's actually caused by a bacterium called Acne vulgaris. And if you suffer from it, you can take one of these drugs and it will clear it up very, very nicely. And so the drugs, the different flavors of these drugs are doxycycline and tetracycline. And they are used, again, very good for respiratory tract infections. And when people come to the hospital and they have pneumonia, it means they didn't get it in the hospital, but they got it out in the community. And so that is referred to as CAP, or community-acquired pneumonia. And again, we have lots of data on what's out there in San Francisco and what's resistant as far as the community of San Francisco and the organisms that cause pneumonia. It's also good for cholera. And look here, H. pylori. Remember, last week we were talking about ulcers, and we used to think that ulcers are caused by stress, but we now know that they are caused by the bacterium H. pylori. And the person who proved that drank a cup of H. pylori, and ho, he got an ulcer. He also got a Nobel Prize, but I'm sure that after that, he took some tetracycline to get rid of the H. pylori, and then he still had the Nobel Prize, but he no longer had H. pylori. So, drugs to the rescue. And indicated here, acne. And so while that is good, this use or the effect of this drug for acne also made us realize something unfortunate about these drugs. And so one of the things about the drugs doxycycline and tetracycline is they have a very high affinity for calcium. And that has several implications. When you are forming your bones, you are needing calcium. And when you are young and you use these drugs, the drugs will bind to calcium and they will deposit in your teeth. And the drugs give you a very gray coloration. So before we knew this, young people were given doxycycline and tetracycline, and then they were developing this gray zone in their teeth. And they couldn't help it. It was just a drug that really wants to bind to calcium. And once it's there, it is permanently bound. And so now we know that, so now we don't give it to young people who are still depositing bone in their teeth. But we definitely, before we knew that, there's a generation, you can still see the gray zone in there. I'll get you in a second. Oh, sorry. Oh, sorry. So I thought it was a question, but we have one of the people here. And it's not uncommon. Secondly, what this means is it has implications for how you take these drugs. When you pick up your prescription, you know, they slap all these stickers on and you're like, oh, they just like to slap stickers on. I really don't need to pay attention to it. But it turns out, and I think that too, but I have learned my lesson, and you do have to pay attention to it. Because now that you know that these drugs bind so avidly to calcium, if you take this pill and you take it with a glass of milk, what happens is the milk has calcium, the drug will bind to the milk, the calcium and the drug will go pretty much straight into the toilet. Nothing will get absorbed. You will not have any benefit from the drug because it's going to stick to the calcium and it's going to not get into your body, but just stick to the calcium and go back out. So if it says don't take with milk, you really shouldn't take it with milk because it makes a huge difference. And so to scare you, at the end here is one example of sort of the graying of the teeth. So if you see somebody like that, maybe they couldn't help it, and it's just a side effect of the drugs, unfortunately. So I am going to leave you with that. You've managed your way through 2 classes, the penicillins and the protein synthesis inhibitors, and you have also answered those 4 questions. So thank you very much, and I'll see you next week. And we have a question.